This week we're carrying out a classic science investigation into how water is transported through plants. To join in and carry out this experiment, you will need some fresh celery, which hasn't been cut, some food colouring, you can use any but blue seems to work the best, some fresh water and some waterproof containers. Jars are ideal because they're nice and heavy and they won't fall over. You will also need a knife or a sharp pair of kitchen scissors. First, we will need to make some solutions of coloured water, and to do this you're just going to use your food colouring. You're going to put quite a lot into a small amount of water. For each of your colours, pour quite a large amount because you need a dark colour into your jar. And add water until it's around 2 centimetres deep. This leaves you with a very, very deeply coloured solution. So we've got 4 very, very deeply coloured solutions of coloured water. Next, take your celery and break the stalks off at the base. Now sort through your celery and you need the stalks that have leaves at the top. The spare ones make for a nice snack. Oh, I don't like celery. Once you've washed the taste of celery out of your mouth, you will need to take each of your stalks and snip off the bottom. This part here, that is all closed up, you need to cut off. We need a nice, fresh, open end. Once you've done that, simply put that into one of your coloured solutions. Do the same for each of your colours. You know, I'm cutting them at an angle, and that means that they're not going to sit flat on the bottom of the jar either. So again, a nice freshly cut end. If you'd like to add a step to your investigation, you could also take one of the stalks that has no leaf, treat it in the same way, and pop that into one of your colours. You can compare that when you have some results and see if it's different. Place your jars somewhere safe, warm and preferably sunny. A windowsill is ideal, and you're going to need to leave those for a few days. Keep checking and see if you can observe any changes. Now we're going to take a look at some that we prepared a few days ago. From this distance, not much has changed, but we're going to take a look at them quite close up. If we take a closer look at our stalk which has no leaves, then on first inspection we can't really see any changes. Up at this end, nothing at all. But if we zoom in to the cut end, you can see a pattern of dots. They're quite dark blue and they follow a very specific pattern. They are the end of vessels, of tubes that travel through the stalk and deliver water and other liquids through the plant. Now if you look at our stalk which does have leaves, you can see that again on first inspection it doesn't look particularly different. If we look down at that end, we get this same pattern of blue dots, but they do seem quite a bit darker. But if we take a very, very close look, can you see at the end of the leaves, there's a little bit of blue colouring. It's quite hard to see on the camera, but up close in real life you can see this pattern. So let's investigate a little bit further. So let's take a closer look and compare and contrast our two plant stalks. Remember, the top plant has no leaves, the bottom one has some fresh healthy leaves. What we're going to do is take a look at the bottom end of each. So you can see some blue dots on this one and quite a strong pattern. and again repeated on this one. But as we move up, the dots become much, much less obvious on the plant which didn't have any stalk. A little bit there, but by the time we get to the fourth section, very, very little colour. With our other plant, you can see still very blue and very, very definite, obvious dots. We'll keep moving up, you can still see them there. By the top of this plant, almost no blue coloration. On this one, you can still see it quite clearly. And again, if you look very closely at the leaves, you can see a little bit of blue coloration right at the very tips. So my question for this week is, how did that colour move? Why is the colour more obvious in this plant, the one which had leaves? Is it because this one's thinner and that one's thicker, or is there something else going on? I'd also like you to make a prediction for the second experiment which I set up. My second challenge to you for this week is to set up the experiment for yourselves but use different colours. Will that make any difference? What is your prediction? Check back next week, we'll have a look at these results and see what we've got, and I'd like to hear about yours. So, happy experimenting everyone.